Thank you. Hi, everybody. Thanks for coming to listen to this. I grew up on a farm. I love to be in a lab, and I love to be in a greenhouse. And this isn't one of those places. So bear with me while I try and give a talk here. We've been working for the last 3 or 4 years on introgressing some interesting characteristics into the Yuki hickory, as was mentioned earlier. And those characteristics are aroma, or we can call it fragrance. You just experienced it if you smelled your jasmine rice. It's one gene trait, and it's the same gene in the jasmine as what we're going to talk about here. And we're also going to talk about trying to get a purple pericarp. And this purple pericarp has a light-colored hull, surprisingly, the dark pericarp, and then if you break the grain of rice open, it's bright white in the middle. So it's a beautiful seed. Let me get going here. Okay. So in nature, genes move and change naturally. We all know that. Bees, pollen, wind is, throwing the pollen around. And nature also makes its own transgenics, if you want to say. Agrobacterium tumefaciens was discovered, and now it's used in bacterial-mediated plant transformations. But that's not what we're doing. We're going to work with manual cross-pollination, and it's just kind of a mechanical way of doing something for nature that it could do itself. And I have brought my equipment if anyone wants to look into this further. So the process of cross-pollination involves actually physically cutting every single spikelet on a panicle of rice. And if you've never counted them, there are quite a few. And then beyond that, pulling out 6 anthers from every single spikelet that you've cut open on all the panicles. And you can see that you want to do this a certain time of day. You want to do this in the morning, right before they're about to have anthesis. So the anthers come out easily. And we do this with a little vacuum emasculator in our lab. So it's a pipette tip. It's a very gentle vacuum. And we can pull all 6 of those anthers right out of each one of those. This is the vacuum emasculator. It is a toolbox with a small motor. And yeah, you take this to a high school and talk about the emasculator. It's fun. But it's just we have instructions for it on the web, and we can actually hook it up and people can play with it if you want. It's kind of an interesting thing.
Yeah, this
thank
system,
you.
yeah, this is the system, and it's transportable, and our blueprint is online for anyone.
Parts lists and things like that. So, okay. So after the plant has been emasculated, you basically have a female plant. And you take that female plant and put it in proximity to a plant that has not been emasculated, whose pollen is coming out. And that's the right stage when the pollen is viable. We put those together and we put a little bag over them. I have a bag, so we actually take this and kind of do that, and we slip them down over. And this is what that looks like on a plant. We have to tie it together so they don't fall apart. And then every 15 to 30 minutes, we have to go in and do that for 2 or 3 hours a day for about 2 to 3 days. So it's a pretty labor-intensive process, but it's really rewarding when you see the beautiful new rice coming that you've helped to create. And you can also see, this is a picture of, again, actually doing, we have these lovely magnifying glasses that we all enjoy wearing to see the spikelets better. But you can see that we have to put the plants at different levels. For instance, the PSA plant, which is the purple parent that we're using in this cross, is this tall in the greenhouse. Doesn't do that in the field. So a Yuki plant is down here. And so we have to stack and make this work for us in the greenhouse in any way we can. We do this by a back cross conversion method. And I know some of you have heard this before, so I will go through it a little quicker than I did the first time. We have a recurrent parent, which in this case we're going to use Yuki Hikari, and a donor parent. That's the one we want to donate a particular characteristic. When you cross those two together, initially we're going to get the F1 seed, the initial offspring, like a child of two people, and exactly half of the DNA comes from each parent. If we cross that progeny to the same recurrent parent, the next time we'll get only a quarter of the progeny, or a quarter of the DNA from the donor parent will be in the progeny. We call that the BC1F1. If we keep going with this, as many generations as we'd like to, next time we get an eighth of the genome being from the donor parent. By the BC3F2, we get only a 16th. That's what you're going to see we have today here, is a 16th of the DNA has come from our donor parents, plants in the field, in the last rows. These are the parents we used, and the one in the center is the Yuki Hikari with the short grain, the temperate japonica. Della is kind of an antique variety from the US. It's not grown too much anymore, but it holds great potential for, it has the aroma gene, and the amount of 2AP, which is the chemical released to produce aroma, is quite high in Della. So that was why we chose this particular parent as the donor. The other cross that we made was to the PSA, which is not a fancy name for anything other than purple sticky aromatic, because it didn't have a name. So it's PSA rice, and comes in a nice golden hull, which is surprising to me and a lot of people. But it's beautiful black underneath.
I'm sorry, quick question.
Yes.
That rice, is that also the black rice? Is it white on the inside?
It is white on the inside.
How does it cook up?
We have not been able to cook it.
It cooks up, well,
in general.
You cook it with the pericarp on, it'll color the water pink and all the rice will look pink. But inside it's perfectly white, so if you were to polish it, it would just cook up as white rice, but you're not going to polish it. You're going to cook it in its purple juice, if you will, and the whole thing will become pink. It's quite lovely. And the pericarp is really hard. So colored pericarp is really impenetrable and really hard. Full of all kinds of health benefits, but hard. So you have to cook it much more than even typical brown rice. And you boil it in like a lot of water and boil it for 40 minutes, and it'll still have kind of a little bit more like American wild rice. It'll have that like slightly hard pericarp, and it'll look quite purple on the outside, and then because it opens a little bit, the water inside will look a little pink. That's good.
Okay. So this was our starting point a few years ago. And I'm going to talk a little bit about an allele. Alleles are variations of a gene, and an offspring always gets one allele from each parent. There are dominant, recessive, and codominant alleles. Some examples: a widow's peak hairline is dominant to a straight hairline in humans. Non-blue eyes are dominant to blue eyes. So a blue-eyed person must have two recessive blue alleles in order to have blue eyes. Blood types, you can be A or B or AB. AB is codominant. Both alleles from each parent are expressed equally. None masks another. You can also have some traits that are gender linked, like male, can't find my keys, and female, I can find my keys. It's become very apparent. I think wheels is also related to males. Tractors and wheels are also male related. Okay. So the purple is a dominant trait. If we have our initial cross and we get our F1 seed, every one of these seeds will appear purple, but they will carry an allele. So they could potentially, when crossed to something else, produce a white pericarp child, offspring seed. This is what that cross actually looked like. And this was a beautiful picture that I love because Gen was gazing lovingly at it in the greenhouse. It was the first time that, again, had made a purple cross, and the F1 was purple, and it was very exciting to us. Aroma, on the other hand, is recessive. So we need 2 doses of a recessive allele in order to get aroma. But all the F1 seeds will carry the potential to cross and become aromatic. This is what our first aromatic cross looked like. So let's continue with the recessive trait down the line here for aroma. We back crossed again to get our BC1 F1s. And at this point, half of the seeds will have no potential to ever be aromatic, while the other half will be heterozygous, and they will have potential to cross and produce aromatic seeds.
But 100% are non-organic.
But 100% are non-aromatic at this point. Okay? Sorry. Okay, so you can't really see aroma. So, and a lot of the plants, some will smell in the field. You can stick your face in some of the aromatic plants and it's lovely, but generally no. So we've been using some marker-assisted selection to help us get rid of some of the plants we don't need to keep because greenhouse costs and labor are high. And this is a agarose gel that we've run a primer, or we've run, a molecular marker that detects whether something has an aroma allele or not. And you can see where I've got a small a, that's the Della. It's fully aromatic. It has 2 recessive alleles, a small a and a small a. The Yuki Hikari has neither. It's only got the large alleles, or large A's, which means it'll never be aromatic. When you see 2 bands, that means it got one from each parent. When you see just the 1 band, you're seeing this, okay, in the progeny here. So the homozygous lines, which means it's inherited both alleles from the same parent, we're going to get rid of all those plants. We don't even transplant them out of their little seedling stage. But we'll keep these plants. And we're going to back cross these again, the next stage, and allow self-fertilization. Once that happens, we don't know ahead of time which ones are going to be aromatic, and we want to get rid of some more plants. So we run some more gels, and we find 25% are fully non-aromatic. They're homozygous, non-aromatic. 50% will be heterozygotes, but at this point we're looking for only something that's aromatic. 25% of these plants will be aromatic, and that's all we keep at this point in time.
And you can determine that how early?
We can determine that when they're about 2 to 3 weeks old.
Before they have seeds, before you would know that they were aromatic.
We plant them in 96 well trays, and we take about maybe 3 millimeters of tissue, and we can get enough DNA to tell whether they're aromatic or not. So why do we combine this lab and greenhouse work with field trials, which we've been doing here with the Yokogis, and very excited to come here and see them this year for the first time. In the greenhouse, we can get up to 3 growing seasons per year if we really work at it. It's a struggle, but we do it, especially with Gen's very good help. Markers allow us to use less space, less money, less time, and we can get the early detection, okay? But we need field trials because plants, and specifically certain genes like aroma, are expressed differently in different environments. Our greenhouse is very tropical, and it's certainly not like growing out in Vermont. So we want to see what they'll do here. And since we can't see aroma, I'm going to pretend and ask you to pretend that aroma is a car, okay? And I have 2 cars. All right. And my lovely electric car does not have the potential to make aroma or exhaust, okay? And my Mustang could make exhaust. When I drive it here nicely, it doesn't. But if I take them to the racetrack. I get exhaust from my Mustang because it had the potential all along to make that exhaust the aroma in a certain environment. My electric car never did. If I put the pedal down, it's still not going to make that nasty smell. Or in the case of plants, that wonderful smell. So by bringing the plants here, or to Maine, or to Pennsylvania, we're hoping we can find the differences in their production. They all have exactly the same gene, but how will the environment affect that aroma? If it's colder, they may produce, when cooked, a more beautiful, more pungent aroma, is the theory. When you grow the plant, actually, the warmer it is, the more pungent it seems to be, the aroma that comes off of the plants. So aroma is expressed to different degrees in different environments, under different stresses, such as temperature. And this rice is now growing outside the tent, like I said. And this is kind of a little mock-up of the field that Ogie has planted so beautifully out there. And the parent rows, as he said earlier, are in the middle. And if we talk about the aroma first, we start with the Yuki Hikari. And the Della, you could see, is a lighter green color. It's bushier, not as erect, and it's certainly not flowering at this point. It's a temperate or a tropical, and it's not really very happy here. The F2 seed, first generation selfed. We can't plant F1 seeds here. There just aren't enough of them, so we have to let things self, and then we generate enough seed for Ogie to do some trials with. So the F2 seed is a mixture. We've got a little different color. Some are a little more spread out, and you can see differences between them. If we go yet another generation, they start to look more and more like the Yuki Hikari. And they're 75% Yuki Hikari at this point in time. And the BC2 F2 generation is 87.5% Yuki Hikari. And they look more and more like the Yuki parent. The last row that you'll see out there is our selected row. So we sent Takeshi and Linda 98 seeds to give a try. They planted them in 72 well trays. And when they were about 4 weeks old, I think it was, they sent me about this much tissue, Federal Express, in a little foamy with some ice. And we got it in the lab, and I extracted some DNA from it and ran some PCR with the Aroma marker and ran it on a gel. And then I'm going to show you some of the plants that we got. This is the actual data from their field on about half of the plants. And you can see the ones that only have the band from Della. We don't want those. The ones that have both bands are still heterozygous, and we don't really want those. But these, at Aroma, have 2 recessive alleles, which is going to cause the plant to have the potential to be aromatic in the field when cooked. There were 21 of these out of 98.
By the way, that's exactly what you're expecting.
Which is exactly what you'd expect, right? The proportion was just about perfect there. And this set on the end, there are 2 rows I think Ogie has that are the selected lines. I think he planted the rest as well just to see, because other traits are segregating in these plants as well. And there might be something else that he likes there. And just because it wasn't what we were looking for in the first place doesn't mean it's a lovely thing that he might like to keep. If we look at the purple pericarp, same thing happens. Plants are different, not nearly as different as I expected them to be. Like I said, in our greenhouse, Yuki is about this tall, and the purple is taller than I am. And I was shocked to see how much smaller the purple plant, parent plant, is in the field. But Ogie has some that he grew in his greenhouse in a bucket, I believe. And they are actually taller. They're still not what we see in the greenhouse, but they're taller than the ones in the field. And the same thing happens here. We get half of the genome is recovered in the second generation, the first generation. In the next generation, we're down to 75%. Here we go to 87.5. And it's interesting, if you do go out to the field and look, you'll see, I think there's 2 plants here that are flowering really early. So you can see some segregation. It's harder to see the physical distinctness of the plants in the field. It's more than I thought. It's more difficult than I thought it would be. But the flowering time is very interesting to see the differences. And finally, in this row, we have our 93.75%. We went through the same process, but the problem here is there are at least 3 genes we found controlling this character. We know what 2 of them are, and we can screen for 2 of them. We are in the process of sequencing and doing some other molecular analyses to try and figure out what that 3rd gene is exactly. So we were only able to tell you, Ogie and Linda, that 2 of these plants have the potential to be purple. We'll see what happens. The others will be a variation from white to brown to really variegated seeds. And I actually brought some of these seeds if anybody would like to see them up here. I mean, these are some of the offspring. We grew 18 in our greenhouse, and you can see this one's just white. And these have some variegated seeds. Okay. Well, there's 18 dishes, sure. I'll pass a few.
So Sandy,
Yes.
you sent them each of these backpacks.
Yes. Yes, and we sent them this as a demo, so we could see how the differences looked in the field. But this is the parent. You can see that one's absolutely black. So let me start these around, and feel free to come up here as well and take a look at some more of those if you want. So in the future, we'd like to continue having Ogie and Linda work with this, and this year they'll be able to make some selections on which of the plants we selected and told them would have both recessive alleles for aroma, and decide if they'd like to carry on all or some of those plants. Ogie tells me there won't be enough to cook and see what it smells like this year. He'll have to plant out more next year to try that. So, and we have never been able to produce enough in our greenhouse to actually cook and smell it. So we're still waiting anxiously for that. Secondly, the purple pericarp lines, we've already advanced them in the greenhouse. Again, has already got them to the BC4 F1 stage. And we're doing this so we have a cleaner background. There's less yuki hikari. And that's important when we're trying to identify a gene, because anything that, more yuki hikari, forgive me, anything that came from the donor parent could potentially be the 3rd gene, I guess I want to say. So we want to get rid of as much as we can and find a purple plant that is mostly yuki hikari background. It makes identification easier, so we kept on going with the back cross there. Next, we need to seek funding to continue our work, which we are in the process of doing. And Genevieve is going to tell you a little bit more about that later on in the day. And I thank you very much for listening.